This video will cover subtopic 3.4 and we'll have a look at observations beyond those of Mendel. So here's the understanding that we'll cover in this video, as well as the applications and skills that we will look at, as well as a bit of guidance. So we'll start out generally here, just kind of looking at testing predictions in crossbreeding experiments. So when we're doing these types of experiments, it turns out that the actual outcomes do not correspond generally with predicted outcomes, or, it can happen. So as we discussed in previous videos, there is a chance, an element of chance with inheritance, being through factors like crossing over or random assortment. So when observations are made, it is important to decide if the results of an experiment are close enough to the predictions for us to accept if they fit, or whether the differences are too great and either the results or the predictions are false. So to determine if the predictions fit the results or the results fit the predictions, we need to use a chi-square test as discussed in the ecology unit. But for the genetics unit, we don't need to do any chi-square tests. So now there were a few problems with Mendel and his observations. Not with Mendel, but with his observations. So in each of Mendel's 7 crosses between different varieties of pea plants, all offspring actually displayed a phenotype similar to mom or dad, so those parental plants. Now, in each of his crosses, one allele was dominant and the other was recessive. So, the dominant allele would be expressed, and you would need 2 copies of the recessive allele to see that particular trait. Now, it turns out in biology, this is not always true. So you don't always have one allele that is dominant to the other. So some gene pairs of alleles have pairs of alleles where both have an effect when they are present together, meaning that, one allele doesn't always mask the other allele. Both alleles can be expressed. And so the 2 forms of this shared expression that we'll look at is one referred to as incomplete dominance and the other one is codominance. With incomplete dominance, this is a condition in which neither allele for a gene completely conceals the presence of the other. It results in an intermediate expression of a trait. So if you want, you can kind of think of it as a blend of the 2 traits, so a mixing of the colors. So in the following example here, we have this example for a type of flower called snapdragons. Now, for the red trait to be expressed, because we are dealing with incomplete dominance, so we're not dealing with one dominant, one recessive allele, we use slightly different notation to indicate this. So, because we are dealing with color, we'll use capital C to represent the trait that we are examining. And then we use a subscript R to represent the color, to represent the specific color of the flower. So in this case, the capital R is red. And then for the other color, which is white, we will represent with a lowercase r, a subscript, a superscript lowercase r. The intermediate variety, or the blending, so when you get heterozygous, which is a combination of the capital R and the lowercase r, you get an intermediate pink color, or a pink color that is generated. So as shown in the diagram here. So in order to get a red color expressed, you need 2 copies of the red allele, and then in order to get the white color, you need 2 copies of the lowercase r. So now if a white flower and a red flower are crossed together, the resulting F1 generation will have the following phenotypes and genotypes to it, so the following appearance. So they will all be heterozygous, where each plant will have a copy of the big R and a copy of the lowercase r. So in this case, this particular genotype results in a pink color for the flower. So we'll have a look at one example of incomplete dominance. So in 4 o'clock flowers, where the gene for the red flower is incompletely, and we're told here in our question that is incompletely dominant over the gene for white flowers, what would be the result of crossing a red flowered plant with a white flowered plant? So we're told that it's incompletely dominant, so we can set up our allele similar to those snapdragons that we looked at on the previous page. So we'll use a C to represent the particular gene. We will use that superscript R to represent the red color. And for a plant to have red flowers, it will need 2 copies of that red allele. And we will define that as being a red flower or a red flowered plant. And the other parental plant that we'll have is one that provides white flowers. And so we'll use our capital C to represent the color, that gene for color, and the superscript lowercase r to represent the white variation of that flower color. Now in this case we are crossing the 2 plants together, so we will indicate that in our cross here. And then to determine the results of this particular cross, we can set up a Punnett square similar to what we did when we looked at monohybrid crosses. So here we'll set up our gametes, or possible gametes for the red flower plant. So one gamete will get 1 allele for the red color, the other gamete will get the other red color, and then we can do the same thing with the white flowered plant. So here are our possible gametes. And then the squares here will represent the combinations of, or the possible combinations of gametes that could join together to create a plant. So here we'll have a big R with a little r, big R with a little r, and it'll all be the same. Make that C look a little bit bigger. And we'll have our genotypes that can result. So from this possible genotype, because we are told that it is the red flower color is incompletely dominant over the white color, we know that we will get a blend of the two, and red plus white will give us a pink color. So we can say that, after a cross, all offspring in the F1 generation will have the following genotype, which will be pink in color. In the next type of shared expression, we have one that's called codominance. So this is a condition in which both alleles for a trait are equally expressed in a heterozygote. So both alleles are dominant. And so the following example here is that we have a roan colored cows have a mix of red and white hairs. So you can have cows that just have this red color and cows that just have this white color. And when you mix the two together, you actually get both alleles expressed. So you get a a mix of the colors here. So you get both the red and the white hairs being expressed. Other examples of codominance include that of sickle cell anemia, where you have your allele for your normal hemoglobin and your allele for your sickle cell hemoglobin, and that if you are homozygous for the sickle cell allele, you will develop the disease of sickle cell anemia. If you are heterozygous for the normal and sickle cell allele, you will get some normal and some sickle cell red blood cells. That's my pen just going a little wonky here. There we go. So these people that have this mix have what's called a heterozygous advantage because they typically do not exhibit signs of sickle cell anemia. So having that one gene for sickle cell anemia is enough to protect them from malaria and allows them to be resistant to malaria, whereas somebody who just has two normal genes is more susceptible to malaria. And if you have two alleles for sickle cell anemia, you are going to develop sickle cell anemia and have this particular genetic disease. So having a blend of the two alleles can provide advantages in certain cases. So in the following example for codominance, we have horses that have a chestnut and white coat colors, and here are the examples here. So here's a horse with that chestnut color. Here's a horse with a white color. And these coat colors are codominant, meaning they will both be expressed in a heterozygous individual. So heterozygous horses express both types of colors, which appear as a golden tan color. So here is that golden tan color where you have both white hairs and chestnut hairs being expressed. And to our eye, we perceive it as a golden tan color. Such heterozygous horses are referred to as palomino or palomino's horses. We're asked to predict the genotypic and phenotypic ratios of a white horse crossed with a palomino's horse. So here we just have the visual representations of what those horses look like. So in the question, we're asked to figure out the genotypic and phenotypic ratios of a white horse crossed with a palomino's horse. So in this case, because they are codominant, we will still express the white allele, or we'll express the allele for color as being a capital C. But because they are both dominant in this case, we will use a capital W, or a W to represent the white color. And we will use a C, a superscript C, to represent the chestnut color. And so a horse that is chestnut in color will have two alleles of that chestnut gene. A white horse will have two alleles for that white color. A palomino horse, which we're told is heterozygous, will have one allele for the chestnut color and one allele for the white color. And so we're asked to determine the ratios, or that F1 generation when we cross a white horse with a palomino's horse here. So we'll set up our Punnett square. Hopefully your lines are a lot straighter than mine. And here we'll have our white horse, and here we'll have our palomino's horse, and then we'll have our possible gametes from the white horse. So those gametes will only have alleles for the white color. The palomino's horse will have a gamete containing an allele for the chestnut color and an allele for the white color. So we look at the possible combinations that can result. Oops. Should be a C. That's thinking W. So there's a C with a superscript W. U and W, and then here is, oops, make that a C. And there's my pen going all crazy again. That is a C with a superscript W. And then our last possible combinations. So when we look at our genotypes and phenotypes that result, so we can see that two horses that have the same genotype, or 50% of the offspring will have the same, will have one particular genotype, 50% will have this other type here. So if they want our ratios here, so we can say 50% will have the following genotype, and in terms of phenotype, it will be a palomino horse, and then the other 50% will only have that white allele, and so it will be white in color. Now, there can be situations where there are more than two alleles for a given trait. So many traits in humans are actually the result of interactions of more than two alleles. In humans, the example would be human blood groups, which we'll have a look at. But other examples that exist out in nature include cloverleaf pattern and rabbit coat colors. You know, rabbits come in a variety of colors. So looking at human blood groups here, so there is a single gene that determines a person's ABO blood type. And that gene determines what antigen proteins or what proteins are attached to the cell membrane of the red blood cells. So that particular gene will control what particular protein is expressed on the surface of the red blood cell. And so a person who doesn't express those proteins, would recognize other types of red blood cells as being foreign and will generate an immune response to them. Now, the gene for blood group is designated as a capital I and has three common alleles. So there is the A allele, the B allele, and then what is called type O, which is just the lowercase I. Now the combinations of these three alleles can produce four different blood types. So if you are type A, you have two copies of the A allele, or one copy of the A allele and one copy of the O allele. If you are type B, you have two copies of the B allele, or one copy of the B allele, one copy of the type O allele. If you are type AB, you have one of the A allele, one of the B allele. And these alleles are equally expressed. So in this case, they would be codominant. So in a red blood cell that has one A allele, one B allele, you would get expression of the A allele and the B allele. Whereas if you were type A, you would just have the A protein expressed on the cell surface, and if you were only type B, you would only have the B protein expressed on the cell surface. Now, somebody who is type O has two copies of the type O allele, and they actually express no proteins on the surface in terms of those particular AB proteins. So they don't have A and they don't have B on the surface of their protein of their cell membrane. Now, in terms of using this to determine who can receive blood, so a person who is type B, for example, can receive only type B blood or type O blood. If a person who is type B receives type A blood, they will recognize this A protein as being foreign and will generate antibodies to it. So a person who is type B cannot receive type A blood. A person who is type B can also not receive type AB blood because these blood cells also express the A protein on their surface. And so that person would react against these blood cells as well. And the same situation occurs if somebody is type A, for example. So just everything that I said before, but with type B blood now present. So type O blood is generally referred to as the universal donor because you can donate to any blood type, and because it doesn't express any of these proteins, it will not be recognized as being foreign. Somebody who is type AB can receive blood from anybody is generally referred to as the universal recipient. So in the following example, for looking at multiple alleles and blood types, so if a man has type O blood and a woman has type B blood, what possible types could their children have? So if the couple has 6 children, all with type B blood, what could you infer about the woman's genotype? So we'll look at the possible genotypes of the man and the woman. So for the man, we're told that he is type O blood. So right away, we know that in order to have type O blood, you need 2 copies of that O or type O gene. For the mother who we're told has type B blood, there are 2 possible combinations of genotypes that the mother could be. So she could have 2 copies of the type B allele, or she could have 1 copy of the type B allele, 1 copy of the type O allele. So it asks what possible blood types could their children have. So in this case, if we consider both types, they could either be type B or they could be type O if the mother is heterozygous for that B blood type. So there are the possible types their children could have. But now we're told that if the couple has 6 children, all with type B blood, what could you infer about the woman's genotype? Now, we'll set up a Punnett square just to kind of look at some of the ratios and probabilities that we would have. And we'll do this using assuming that the mother is heterozygous for the type B blood. So we'll set up our Punnett square for the father, and then for the mother, and then we'll look at the possible genotypes that the children could have. So here we see that there are the two possible genotypes that the children can have. Um, we can expect that there is a 50% chance that the child could be type B, and a 50% chance that the child could be type O. So the fact that they've had 6 children and all of them have type B blood strongly suggests that the mom, the genotype of the mom, is most likely homozygous for the type B allele. There's a higher probability of that, um, given the fact that if she were type B, all of the children could only ever be type B, and the fact that there's been 6 children, all of them with type B, points to a high likelihood that she is this particular genotype. Although, there is a small chance that she could be um, heterozygous, and it just so happens that every time, for all 6 children, they've ended up being type B blood. Um, Small chance, but is probable. But we'll go with the higher likelihood that she is homozygous for that type B allele. So that is the end of this video for 3.4.